Hey everyone, welcome to the 5th video in our series for AP Review. This one focuses on the topics of meiosis and crossing over. So with meiosis, the key thing to remember is that this will only occur in gametes, which are the sex cells. And the biggest difference between meiosis and mitosis is that there are 2 rounds of division in meiosis, and that's going to cut the number of chromosomes in half. In other words, a cell is going to go from diploid down to haploid, so that when fertilization occurs, the number of chromosomes in the new individual formed is back to diploid. So in the case of humans, we have 46 total chromosomes, so our diploid number, or 2N, is 46, and then the haploid number for 23. So the sex chromosomes, or the sex cells, will have 23 chromosomes each, and then when a gamete fuses with another gamete, it's going to create an individual with a full set of chromosomes, which is your diploid number. In the case of meiosis, the processes themselves are very similar to the cell cycle. One of the things to remember, though, is that you have homologous chromosomes line up during meiosis 1, and then in meiosis 2, those homologous chromosomes are going to be split. So because the homologous chromosomes pair up during meiosis 1, something very important happens during prophase 1, and that is the concept of crossing over. So crossing over is going to increase your genetic variability and increase the chances that the organisms are going to be much different, or the gametes that are produced are much different than what would be possible to produce if crossing over did not occur. Now there are some exceptions to this. So some of the exceptions that we talked about include linked genes, and those linked genes, what happens is, because the traits are physically close together, they don't separate the way that you would predict using traditional Mendelian genetics. So they are physically close together on the chromosome, and your inheritance patterns are different than what you would expect if you followed traditional Punnett squares. The example that we talked about with linked genes are red hair and freckles. But otherwise, during meiosis, you have those 2 rounds of division. We did talk about one additional thing, which was nondisjunction. And that is when the chromosomes fail to separate properly. With nondisjunction, you can have either an additional chromosome, so that would be 2N plus 1, such as with trisomy, or you could have one missing. This is 2N minus 1, which is considered monosomy. You also have some nondisjunction that occurs in the sex chromosome. We talked about Klinefelter-Turner syndrome. But those are all examples of what happens when meiosis doesn't occur correctly. As far as meiosis goes, don't worry about the details of what's happening in each stage. You do need to know that they're the 2 rounds of division, and that there's not an extra synthesis phase in between. You do need to know that it occurs in those sex cells, your gametes, and that during prophase 1 there is the additional process of crossing over that is going to take place. I hope this helps you guys. Good luck studying.